Case Western Reserve University's Institute for the Science of Origins proudly presents the Origin Science Scholars Program. The Institute advances the scientific understanding and application of origins and evolution of human and natural systems. The Origin Science Scholars Lectures are presented with the assistance of Case Western Reserve University's Siegel Lifelong Learning Program, College of Arts and Sciences, and Media Vision.
Tonight, it's my pleasure to introduce Peter Hart. Dr. Hart is professor of genetics at Case Western Reserve University. He received his PhD in biology from Yale University for work on the genetic control of visual system development in fruit flies. Then went on to the Department of Biochemistry at Stanford University, where he characterized the homeotic gene ultrabithorax before joining CWRU in 1986. His research interests center around the molecular mechanisms underlying regulation of homeotic gene expression during Drosophila development. Figuring out how fruit fly genes work to produce fruit fly bodies doesn't just benefit fruit flies. It gives vital information on human development and how all life on Earth has evolved. Let's welcome Professor Hart.
It's a pleasure to be here, and I'm going to talk to you tonight about the background for the next two sets of lectures on sleep regulation by circadian rhythms. So I'm going to restrict my attention to the discovery of the underlying molecular mechanisms of the clock, which, by the way, are conserved in humans with minor variations, and in fact are conserved throughout the kingdoms of life. So here's a little spoiler. The first slide shows you the mechanism that we're going to unravel, and I'm going to tell you how we got there. So this is to remind you all that we live on the planet Earth, and everything that I'm about to say is due to the rotation of the Earth, since circadian rhythms are, in fact, an adaptation to life on Earth. So most of you know from personal experience that we have daily rhythms that regulate much of our physiology. In fact, much more than we appreciate. In fact, so-called chronobiology has become a regular cottage industry, and it's discovering more and more ways in which the daily light-dark cycle regulates our physiology and behavior. I only put some of the most salient ones that we've known about for a long time up here, which include regulation of blood pressure, temperature, secretion of hormones, including melatonin, which influences the circadian rhythms themselves. Many, many other aspects of our physiology. And of course, sleep and activity when we're awake. We have periods when we're not doing much, we take a rest, and periods when we're very active and alert. So let's just get some definitions of terms out there. The word circadian comes from the Latin circa, meaning about, and dies, meaning day. It refers to cycles and rhythms with periods of about a day, by which I mean approximately a day. Most of the rhythms are about 24 hours, but they are tuned by subtle regulatory mechanisms that synchronize the phase of the circadian rhythm to the light-dark cycle, and we'll talk a little bit about that later. Circadian rhythms are intrinsic oscillations in behavior and physiology. That is, there's an internal clock, as we'll see, and it's self-sustaining and autonomous. It allows the organism's body to unconsciously anticipate the changes in the environment that are due to the light-dark cycle. And there's a great quote from the recent Nobel laureate, Michael Young, who says, in a sense, rhythmic physiological anticipation can be thought of as a primitive mechanism of planning and cognition. That's something to think about. It's actually a profoundly interesting and provocative thought, to think that much of what we do is really things that we aren't aware of. So importantly, circadian rhythms are found in all kingdoms of life on Earth. That includes animals, plants, fungi, and everything else, which includes bacteria. I don't think to my knowledge, we've looked in archaeobacteria, but certainly eubacteria, which are the ones that we used to know all about before they discovered archaeobacteria, we know some of them have been shown to have circadian rhythms. This means that these are ancient, and it means because we all came from a common ancestor, it means that they originated very, very early after the appearance of life on Earth. And it's also potentially means that they could be conserved mechanisms operating underlying all of them. So one of the early debates, and this goes back almost 300 years, was, well, we see these behaviors of organisms. Are they due to an internal clock? And one of the stars of the early experiments was the Mimosa plant, which many of you know opens its leaves in the morning to the light and closes them at dusk. And a very important early experiment was done, which basically addressed the question of whether there was an internal self-sustaining clock. When the Mimosa plant is placed in continuous dark, the 24-hour period, that is the length of the rhythm, persists in the dark. So, for example, the plant in complete dark remembers to open at the time when the local day begins. And this I mean, they didn't do this for too long, but it persists for many days. The interesting thing, which was also observed, was that the phase, which is its synchronization with the day, drifts, because in the total dark, it drifts. And this suggested that there's a role for light in setting or synchronizing the phase of the 24-hour period with the local daytime. And this experiment was carried out by an astronomer in 1729. So it's been a long time that we've known or suspected that there's a biological clock. And by now, it's very well established that this is an internal clock that is self-sustaining and entrains many of the other rhythms in our body. So just last year, the Nobel Prize in Medicine was awarded to 3 Drosophila geneticists who used the Drosophila experimental system to unravel the nature of the clock. This effort spanned 3 decades, and the 2 fellows on the left, Hall and Rosbash, collaborated, and they competed with Michael Young. And believe me, Michael Young held his own. So the star of the show for them was Drosophila. And I guess it's fair to say that the contribution of Drosophila was, not to be modest, was discovering that the biological clock's molecular mechanism is genetically encoded, which seems obvious to us now, but was really a bone of contention all through the 60s and 70s. And also showed that it was evolutionarily conserved, because once the genes that we're about to talk about were isolated, they were immediately taken and used to identify the human genes and the mouse genes, and they've been found in many other organisms now. And the mechanism, I can say, as a spoiler alert, is conserved in many of the organisms that have been examined, with minor and interesting variations on the theme. So one of the things I'd like you to learn today, in addition to the facts of the matter, is how geneticists think. Because the remarkable thing about this effort, which involved a significant amount of biochemistry to figure out how the molecular mechanism works, really started by isolating mutations. Every single one of the core clock components was first identified by a single mutation in a gene that was then isolated, and the biochemistry was exercised on the protein product of that gene in order to understand how it worked and how they all work together to create a mechanism. So the great thing about this approach is that it requires absolutely no knowledge going into the problem of how the system works, right? If you're a biochemist, you might want to understand something about how something in the blood works, and you might try to purify something from the blood. But behavior and rhythms are what we call emergent phenotypes, a genetic-y word. And emergent phenotypes are difficult to imagine where to begin. But for a geneticist, it's quite simple. You just ask for mutants. You ask the organism to give you mutants that perturb the biological problem of interest. Now, this is not trivial, but let me just go through another reason for why we take this approach. Why do we want to get the DNA? And why do we want the mutants? Well, the mutants tell us that we have a gene because mutations occur in genes. And the mutation allows us to map the genes to DNA, to a position on the chromosomes. And the DNA is then isolated from the region where the mutation maps, and we'll see how the gene is identified. But remember that there's and for some of you this will be the first time you hear it, the so-called central dogma of molecular biology, sometimes abbreviated as DNA makes RNA makes protein, tells us that DNA is transcribed into messenger RNA, a single-stranded copy of one of the strands of the DNA. And although some RNAs act on their own, most of the ones that do the business of the cell are translated, as is said, into protein by the ribosomes. And you can think of the ribosome as a 3D printer because it prints out a linear polymer of the 20 amino acids in a order that is dictated by the sequence of the gene. So proteins have all have unique sequences, and the sequence dictates the shape that it folds into, and the 3D structure is what allows it to do its work. So we want to get our hands on the DNA so that we can program bacteria to make lots of protein to study how the protein works by biochemistry.
I'm familiar with the adenine, the ATGC, but I am not familiar with the U. What is the uracil?
Yes.
I see it's in the RNA. It replaces...
Yes, it replaces thymine, or the...
Why does it...
Why?
Yeah, or how does it do that, I guess?
You'd have to ask the maker.
Sometimes things really are just historical accident.
Well,
Once
of course.
the system gets going, it continues going unless it's really not good enough to work. Once you get a toehold, it's just, contingencies of history very often. There may be mechanistic reasons, but nobody has suggested any, right?
What characteristics of Drosophila sleep allow you to pick out mutant genes, to know which DNA you want to?
Well, we're not going to talk about sleep today. It was actually a different rhythm, and we'll see in a minute how that was done. It was actually using an activity rhythm, which is very robust. So I'll show you in a minute exactly how they designed the genetic screen, as it's called, to isolate those mutants. But you put your finger on the real trick in genetics is designing a good screen, because if it's really robust and you can easily distinguish the mutants from the normals, you usually don't get a lot of background garbage. You don't always get what you think you're going to get, but it's amazingly successful at finding things eventually.
Thank you for joining us. You've been watching Dr. Peter Hart discussing circadian rhythms, the daily clocks within biological organisms. For more information on the Origin Science Scholars Program, please visit the Institute's website at origins.case.edu. In the next part of the talk, Dr. Hart will discuss the discovery in fruit flies of the period gene, which underlies circadian rhythm. Now, back to the talk.
We're going to talk about 6 mutants that led to the core components of the clock. And we're going to talk about the properties of those and how they act together. And the 6 are period and timeless. You notice that the gene names are abbreviated in lowercase. The proteins are abbreviated in uppercase just to distinguish between them. So the proteins are represented here as the circles and the squares. Now, this is the abstract from the very first paper that identified a mutation in the so-called period or PER gene, which basically opened the door to this whole field. This is an astonishing paper for a number of reasons, one of which is the abstract is among the shortest you'll ever see in the proceedings of the National Academy of Science. And it is an amazingly modest one because it simply states something that is almost unbelievable. So they isolated 3 mutants. One of the mutants is arrhythmic. It has no rhythm. It's random. It does things at random. It's active at random times during the day. Another has a shorter period of 19 hours, and another has a longer period. They used 2 assays. One was looking at, there's a rhythm to how the insect hatches from the pupa or the chrysalis. It happens in the early morning, mostly to evade predators. And they also used a more robust activity or locomotor activity rhythm in which, and I'll show you a second how they do that. The most astonishing thing so they got 3 different flavors of mutations from one screen, and if I tell you how many flies they had to screen, you won't believe it. It was 271 to get 3 mutations. That never happens. Now, the most astonishing thing, though, is they got 3 mutants, and all of them appear to be in the same gene. Now, I'll tell you one other fact that maybe we'll get back to later, which came after this work, and that is that flies that overproduce the PER protein have a shorter rhythm. They overproduce the normal one, so you put in a so-called transgene that allows you to make more PER protein. And if you reduce the amount of PER protein, the normal protein, by taking away 1 copy of normally 2 copies of the gene, they have a longer period. So there's something quantitative about the amount of protein that is relevant. Okay, so here are the 2 characters. Seymour Benzer was a distinguished senior scientist who was fighting a dogma, basically, that had developed in people who were interested in behavior, who thought that it could not be possible to isolate a mutation in a single gene that would have a significant impact on behavior. And Benzer basically felt that he was going to prove these people wrong, which he did in spades. And this is Ron Konopka, his graduate student, I think rather late in his graduate career, perhaps after the publication of the paper. And they designed, so most mutations in Drosophila that were isolated in the first 50 years from around 1903, when Drosophila was first brought into the laboratory to use as an object of study, most of the mutations that were isolated were spontaneous, and they affected anatomical features that were obvious. This was the first mutant, the white eye on the right. Now, behavior is a very different thing than that. And to identify mutations that perturb circadian rhythms, one of the things they did, and I'm only going to talk about the locomotor activity rhythm, is they designed basically an automated way to screen single flies to see if they had normal or abnormal rhythms. So the drill is basically you want to produce more mutations. So it's not done by looking for spontaneous mutants. It's done by feeding flies a mutagen. And although it was first shown in the 1930s, well, 1920s, that by Hermann Muller, that X-rays could cause mutations. They cause very messy mutations that rearrange the DNA and so on. By the 1960s, there were very efficient chemical mutagens. The most popular one was ethyl methane sulfate, which produces mainly point mutations, changing a base or something like that in the DNA. You feed them the mutagen, and then you screen their progeny. And in this case, you can actually screen them individually because it's a very robust phenotype to screen. You screen them for perturbations of their rhythm. Now, the rhythm is how much they move around. So flies like us have a consolidated sleep period, and they have a day period during which they take naps. Now, this is not a very good representation precisely on the right, but it gives you the idea. So the numbers on the y-axis are just the days. And this is a little complicated to read because they repeat the the last reading on one row in the next row. So you can see on the top row, the right-hand pattern is repeated at the beginning of the second. If you just scan down the middle row, you get the idea that it's a fairly robust rhythm. It's not really the same as it looks in the real data, though. And these are 4 flies, a normal fly used as a control, an arrhythmic mutant, a short period mutant, and the long period mutant, the 3 permutants. Now, it's not ideal. There are small interruptions here and there, but you can see large consolidated periods of no activity, where there's no black mark for when the fly crosses the infrared beam. So one thing I forgot to say is that there's an infrared beam that is put across this vial, and when the fly, anytime it walks back and forth across that beam, it registers a tick that is recorded as an activity. And it's fed into a computer, and it can be analyzed directly in real time. So notice that the arrhythmic mutant in particular has, in the second line, has a random activity pattern. There are periods of rest and so on, but they're not consolidated the way they are in the normal, where you have the straight line with no ticks on it. And the short period rhythm has quite consolidated periods of no activity and activity, but the period is shortened between them. And the long period mutant has, as you can see, a longer period, still has consolidated periods of rest. And there's more information in this data. And this was the early days. There are now more sophisticated ways of doing this that have made it even more robust. Virtually all the mutants that we're going to talk about have come from this screen that isolated the components of the clock. So once the mutants were established, remember, we get the mutants. We make sure that they have a robust phenotype that shows perturbation of the locomotor rhythm. Then we map them. So having a mutation allows you to use classical genetics by doing genetic crosses like most of us had in high school when we studied Mendel. You can now precisely localize where a mutation, and therefore the gene that encodes it, where that mutation is on the chromosomes. And that you can narrow it down now to very small regions, which make it quite straightforward to isolate the DNA that covers that region. And this is exactly what happened in the case of the per, or period gene. The top line with the numbers represents the DNA, the chromosomal DNA. And you can see the span of this from about 2680K to 2700K means 2,680,000 base pairs. That sounds like a lot. It's not. The genome is 170 billion 170 million base pairs. Now, what's interesting is this is quite a small region. So they had narrowed it down well enough that there were only 4 genes that were contained in this region. This is actually a little bit bigger than the region. But the region spans roughly from 2680 to 2700, as shown on this map. So the DNA was isolated by recombinant DNA methods. Nowadays, since we have the entire sequence of many organisms, including Drosophila, you don't have to go get the DNA. You just have to find out where it is, and then you can mail to a company that sends you the DNA. But many, many mutations had been isolated in this region, but no one realized that there was one that affected circadian rhythms. And in fact, a very interesting thing about this mutation, even when you delete the DNA for this gene, it's viable, and it has no visible anatomical phenotype. You would never suspect it was abnormal until you did this activity assay. So it escaped detection of the other mutagenesis of this region because the screens that were done all looked for lethal mutations and ones that had very pronounced anatomical phenotypes. So they had this, there were 4 genes in it. How did they figure out which one was the gene? Well, they took each segment of DNA that contained one of the genes, and they put it back into the genome of the arrhythmic permutant fly. And lucky for them, only one of the fragments of DNA rescued the arrhythmic phenotype and restored the 24-hour rhythm. So that was absolutely the gold standard proof that that piece of DNA included the gene, the PER gene. As a further confirmation, they sequenced the gene in all of the mutants, the 3 PER mutants, and every single one of them had a mutation in the sequence of that gene. So that together with the rescue of the phenotype by the piece of DNA was absolute proof that this was the PER gene. So this is basically the paradigm for how you go from mutation to having the DNA. And as we said before, that provides you access to large bucket loads of the protein to do biochemistry on. So when the sequence of the PER protein, which you can predict from the sequence of the messenger RNA or the DNA, when the sequence was inspected, it gave no clue. It wasn't similar to any other genes that had been isolated. And so that wasn't very helpful. So the understanding what it did was going to have to wait until we had more information, probably for biochemistry. But an amazing thing that suggested right away that this was a key component of the central oscillator of the circadian clock was that the RNA, and remember we talked about how the DNA is transcribed by RNA polymerase into a messenger RNA. The levels or the abundance of the RNA varied throughout the day on a 24-hour cycle. So it had the same period as the behaviors that the mutant had perturbed. That was amazing. And the protein, which is translated from the mRNA, is also, not surprisingly, exhibiting a circadian oscillation over the course of the day. On the bottom, you can see the light-dark cycle indicated by the white and black bars. And you can see that the PER protein accumulation over time is offset by about 4 hours from the accumulation of the messenger RNA. I'll tell you now that this is not simply due to the time it takes to translate the RNA. There's another little trick that's going on here that we'll talk about in a few minutes, which is there's a delay mechanism built into the clock to allow the period of the cycle to stretch a little bit or shrink to accommodate seasonal changes in day length. And that's a theme that is still being worked out by discovering new mutations and other things that do more fine-tuning of the clock. Now, the other surprise was that in the PER mutant, the arrhythmic mutant, the RNA oscillation is abolished. But the RNA wasn't abolished. It actually stayed constantly at the high level that is illustrated by near the peak of the messenger RNA. So it suggests that the PER protein is somehow inhibiting its own, the RNA from its own gene that encodes it. And remarkably, the arrhythmic mutant among the 3 is the only one that produces no protein. It's a special kind of mutant that truncates the protein, and basically truncated proteins are most often disposed of very rapidly, and sometimes you can't even detect them. So this was an amazing number of insights from a couple of experiments. It was surprising that the first mutant had oscillation in its RNA protein levels that matched the period of the actual behavior, the 24-hour rhythm. And it was a good hint that this is a good candidate for a core clock mechanism. There are a few other things. So just as a summary, for emphasis, it revealed that the oscillation, in particular, is essential for the behavioral rhythms. And the fact that the PER arrhythmic mutant made no protein revealed that the PER protein was essential for the oscillation, right? Suggests that it might regulate its own gene. And of course, it suggests somehow, by a yet-to-be-discovered means, that the per protein represses its own transcription and induces oscillation. And we'll see exactly how that last part occurs.
When you showed the activity of the different mutants, it looked to me like, in fact, the difference in the cycle is simply due to the length of time that they're active, that in fact the resting periods is about the same in all of them, and it's just changing that activity period, the length of activity period. So that seems to be that's an interesting difference.
Yes, that did not escape my attention, and it's a very interesting observation. I have no idea why yet, but I think we might hear from some of the sleep experts next week as to how this might happen. Yeah, there's a lot of information in this data, and it's not all been plumbed in this paper, for sure.
I'm curious about possible temperature variations and what that would mean in terms of the oscillator rhythms.
One of the well-known characteristics or features of a central oscillator or circadian clock is that it's so-called temperature compensated. And however, that doesn't mean that the clock is unaffected by temperature. It just means that the period is refractory to the effects of temperature. However, and this is a bee's nest that we don't want to get into right now, but in fact, temperature is a highly correlated circadian rhythm that tracks the light, right? Because it's hotter during the day. No matter what the average daily temperature is, it's hotter during the day than it is at night, unless there's a heat wave that comes in. The light provides the energy that affects the temperature. So temperature is actually one of the physical variables that has to be dealt with by circadian rhythms, and adapting to the temperature changes on the circadian cycle is something that the clock also influences, for sure. But the clock period is temperature compensated in a way that we really can't get into right now. There's so much we don't know about this, even though we understand the core mechanism quite well. And the fine-tuning of it continues to spill out in publications to this day.
I'm about a half a generation too old to understand molecular DNA experiments. And the question is, after you've spliced the mutant back into the genome, how do you reconstruct the fly so you can do the test? The other question is, why are we looking for 24 hours when the day length at the time of evolution of animals might have been more like 21 or 22 hours, with the Earth's rotation having slowed in the intermediate period?
I guess I'm free to say that I wasn't there, and it probably was entrained to a 21-hour day and evolved. And in fact, one of the interesting things about the per protein is one of the investigators, Michael Rosbash, fiddled around with deleting parts of the middle. There's a repeated sequence in the middle. I can't remember which two amino acids, but it's a repeat of two amino acids over and over and over again. And he noticed that in different species, it was different lengths. So he started deleting parts of it, and he found that the period length was affected by varying size deletions of parts of that amino acid repeat. So, the clock evolved itself, and it continues to evolve. And how about organisms that live at the poles? They're experiencing something quite different. Yeah, the rest of us move around a lot, but a lot of organisms don't move around a lot, especially plants.
We hope you've been enjoying the Origin Science Scholars Program with Dr. Peter Hart. Dr. Hart is Professor of Genetics at Case Western Reserve University and a Fellow of the Institute for the Science of Origins. In the second part of our talk, we learned how scientists use mutant fruit flies with anomalous circadian rhythms to discover the genetic driver of the fruit fly's internal clock. In our final segment, Dr. Hart will talk about how this original observation has been expanded on and extended to mammalian clock genes. Now, back to our talk.
It was almost, well, it was a long time. I mean, the PER mutants were actually isolated in the late 60s before they were published. So another mutant wasn't isolated until 1994, and it was called Timeless. Now, there's not much to say about Timeless because it behaves exactly like PER. Its RNA and protein levels oscillate. Here's what's interesting, though. In the Timeless mutants, the PER mRNA oscillation is abolished, and vice versa. So these two things look like they're doing the same thing. And it also suggests that both of them are required for the oscillation of both mRNAs. And a little bit of biochemistry answered the question that was pretty obvious, which is, do these proteins cooperate in some way? i.e., among other possibilities, do they get together and form a dimer? And the answer to that was yes. That was the first biochemistry experiment that gave some insight into the mechanism, and you'll see why in a minute. So you can lay the curves right over one another. The RNAs on the left, the gray lines are the two mRNAs, the two proteins. They track each other. Now, this is not surprising if both the PER and TIM proteins get together and regulate the expression of both the TIM and the PER genes. So here is a summary model so far of what we know. So the arrow sitting on the DNA indicates where RNA polymerase begins transcription of the messenger RNA, that is the single-stranded little braid that's coming off the gene. Now, RNA has to go out into the cytoplasm, so the small circle is the nucleus, and it goes out into the cytoplasm to be translated because the ribosomes are out there. And the ribosomes generate the amino acid polymer that defines the PER protein sequence, and then that polymer folds into a complex structure that is unique to the sequence. The sequence information, the protein sequence, determines the structure of the protein, the 3D structure. So every protein is different in that respect, depending on the differences in its sequence. And likewise, the TIM protein, this applies to either of the genes. Now, one of the things that was apparent when we had ways to detect the proteins was that the PER protein didn't go back into the nucleus right away, although it did wind up there eventually. But it accumulates in the cytoplasm until there's a significant amount of it. And it becomes a heterodimer with TIM when both proteins have a high enough concentration to drive dimerization. They have to find each other to form heterodimers. And it turns out that the PER protein is somewhat unstable. And so the longer it sits around not bound to TIM, it's prone to degradation. And there's a whole machinery in the cell that takes care of that very efficiently. But once it's bound to TIM, the heterodimer is very stable. Both proteins further stabilize each other's lifetime. And every protein has a half-life that is characteristic of it, left to its own devices. But there are also mechanisms by which you can direct things to the trash heap by other proteins that can interact with it. And we'll see an example of that in a minute. So there's a delay that we saw in the way the protein and the mRNA accumulate. The mRNA, of course, preceding the protein with a lag of about 4 hours between the two peaks. So once in the nucleus, which is good news for the model suggesting that the PER and TIM proteins regulate their own transcription, the transcription of their own genes, because you have to be in the nucleus where the chromosomes are to do that. And so neither of these proteins have sequences that reveal anything about what they might be doing. Most molecular biologists pray for a gene, a brand new gene that they isolate that looks like another gene in some way. So for example, it has a DNA binding domain. That's Eureka. Crystallographers don't like that because they like new proteins, but you can learn things from the sequence if it has something similar to a well-known protein. And these two proteins are completely unique. They have no features about their sequence that revealed anything. So that will have to wait. And we still don't know how these genes are turned on. So usually, genes are inactive. They must be actively activated by proteins known as transcription factors that bind near their start of transcription. And so here they are. There are two other proteins encoded by the clock and cycle genes, which were identified by arrhythmic mutants with properties similar to PER and TIM. But these guys, actually, the sequence was the jackpot. They both have a sequence, a protein motif in them that is well known in a whole class of transcription factors that bind directly to a specific DNA sequence, or bind directly to DNA sequences. And they also form a heterodimer, and the sequence that they bind to is found in the PER and TIM genes, abbreviated CAC-GTG, 6 bases. And they're transcriptional activators. So it adds another element to the picture. And furthermore, although PER and TIM have no DNA binding activity, that is explained by the fact that it was discovered through biochemistry that the PER and TIM heterodimer binds directly to the clock cycle heterodimer and somehow blocks its ability to activate the genes. Okay, so that essentially is the minimal machinery needed to have a self-sustaining, oscillating transcriptional machine. Now, I want to mention one other. Remember, we talked about the fact that the PER protein kind of hangs around on its own in the cytoplasm until its level builds up. And its level is determined, of course, by this new synthesis of RNAs that can be translated from the gene. But there's another wrinkle here, which is a mutant called double time. And the reason is because it has a shorter period. So it's unique among the mutants so far in that the other genes tended to mutate to arrhythmic conditions. And you'll see why in a second. The double time protein actually encodes a well-known enzyme, which was completely unknown to have anything to do with circadian rhythms. It's called Casein Kinase 2, because a biochemist once threw it in a tube with some casein isolated from milk and showed that it phosphorylated it. Doesn't mean that's what it does in vivo, but it phosphorylates lots of proteins. And it turns out the PER protein is one of the proteins it phosphorylates. That is, it adds a phosphate group to several of the amino acid side chains that comprise the PER protein sequence. And that causes the PER protein to have a shorter half-life. In fact, it gets captured by the protein degradation machinery and it's eliminated. So this actually represents another wrinkle in that it introduces a delay mechanism into the clock that can be exploited by varying the level of the CK1 enzyme or other. It turns out it phosphorylates a number of sites on the protein, and a number of sites affects how rapidly it's degraded. So we have a timer now, a self-sustaining oscillator, that has at least one example, and there are multiple other ones that we're not going to talk about, but it's a good example of how you can have a delay mechanism that can itself be potentially regulated to give you some flexibility in expanding and contracting the period of the oscillator. And I think I mentioned that when PER and TIM, and likewise when clock and cycle form heterodimers, it greatly stabilizes both proteins in the heterodimer. Most of you are probably wondering, yeah, okay, you've got something that can regulate itself and it has a period of about 24 hours, but how the heck does it get synchronized with the day? And remember, the key feature is that it's self-sustaining, period. But that doesn't answer the question of how it gets entrained to the light-dark cycle. So there is a protein that was also discovered by a mutation called Cryptochrome, and it's the primary light sensor in the system. And Cryptochrome synchronizes the oscillator to the light-dark cycle. And how it does this is interesting because what is it? To be the light sensor, it has to do something in the light that it doesn't do in the dark. And what it does is the energy from the light changes the shape of the protein and makes it able to bind to the TIM protein. And when it does that, the TIM protein is degraded rapidly. Now remember, I said the TIM protein is stabilizing PER. So when you get rid of TIM, the PER protein is also degraded. Now this all takes some time, which is why we have the slow, it's not a step function. You get a slow decay of the proteins and the mRNAs as well. So here is a summary of the central oscillator. And I'm going to test you on what you think we haven't answered yet, because there's one big question that we haven't answered yet, and I'm sure it's occurred to some of you. So we have the PER and TIM genes bound by clock in the absence of binding by when PER and TIM are not available, namely during the day. We have the clock and cycle heterodimer driving the expression of the PER and TIM genes, making mRNA that it exports out to the cytoplasm. It gets translated in the cytoplasm and gradually accumulates as more and more is translated. But competing with the accumulation of it based only on the production of mRNA is the competing process of the double-time protein phosphorylating it and attenuating the level, right? But enough of it accumulates eventually, and likewise for TIM, which is not subject to the phosphorylation by double time. Enough of it accumulates that they eventually form heterodimers, and they go in and it binds to the clock cycle proteins and inhibits its ability of that heterodimer to activate transcription of more per RNA. So you can see how this can create an oscillation, right? Your first thought is, well, it binds to clock and cycle, game over, the clock stops. Well, yes and no, because what happens when you inhibit is that you stop making mRNA. So now the half-life of the proteins PER and TIM are partly the determinants of how long it takes for the existing PER and TIM proteins to turn over and go away. And that is actually helped by the cryptochrome protein, because the cryptochrome protein is out in the cytoplasm. It binds to TIM as soon as it accumulates enough light. And it's a very interesting process because it can integrate very faint light over about 6 hours. So it probably starts changing shape, unless there's a threshold, as soon as, before sunrise, when you start to get dim light. And so it's very sensitive to the light. But once the shape changes and it binds to TIM, TIM is cleared very rapidly. And then PER is degraded. So once that happens, you've freed up clock and cycle to initiate RNA synthesis again. So the length of the period, of course, is regulated by a number of the variables that we talked about, right? How long it takes to accumulate enough PER, what's the half-life of PER and TIM, and so on. So we have what looks like something that can be self-sustaining. It regulates it. The clock regulates. So this is the core clock, the core oscillator in the organism. And Cry provides the way to constantly readjust things so that you have the period entrained to the onset of light. It's a minor adjustment every day unless you travel, but it assures that you keep things in sync. So we travel at our own risk because there's all kinds of desynchronization that happens in your other in many different tissues. I think we talked enough about this. Oops. I'm going backwards. What is this? Yes. So some of you surely have wondered, gee, you got a great mechanism for a couple of proteins to regulate their own transcription. That's really neat. Self-sustaining and all that. But how the heck does that control everything else that it's known to influence? How does it hook up with all the rest of the physiological processes that the central clock is alleged to be controlling? And the answer is quite simple if you think about it for a moment, because clock and cycle regulate the per and tim genes. Per and tim and clock and cycle are present in most, if not all, cells in the body. So they're available to regulate other genes. But what you need to do is you need to get clock and cycle to participate in the regulation of those other genes. And it turns out a number of recent studies, and most of this stuff is very recent, people have suspected it for a long time, and there have been examples of other genes outside of the central clock that are regulated by clock and cycle. If you inspect the whole sequence of the genome, if you look in the vicinity of all of the genes, there are about 20,000 genes in flies and roughly the same number in humans. The number fluctuates between 20 and 22,000. A lot of people thought there were going to be 100,000 in humans because, after all, we're human, we're very complicated. That seems not to be the case. But it turns out that their clock and cycle binding sites, that CAC GTG DNA sequence, in front of lots of genes, thousands of genes. Now, the presence of the sequence does not guarantee that the proteins are going to bind there, even if they're in the cell. There are a lot of contextual effects that can overrule the simple correlation between the presence of the sequence and the binding of the protein that recognizes it. However, there have been two recent studies that have shown two really important properties of these genes, many of them. And it is thousands. It may be half of the genes in our bodies. There's actually a recent paper on baboons that claims it's 80% of our genes. So two things have been shown. Many, many, many of these genes have oscillating mRNA levels. How do we discover that? Well, it turns out nowadays you can actually isolate all of the messenger RNAs in bulk from the cells. And you can sequence them. And every sequence reaction that creates a bit of an RNA sequence identifies the gene because it has a unique sequence, and it's a count. So you can watch by sampling from tissues, and this has been done for lots of different tissues, every 15 minutes, every hour, or whatever, however much patience you have. And you find that many of these genes indeed have mRNAs that oscillate just like per and tim over the course of a 24-hour period. So that suggests that they are at least somehow directly or indirectly regulated. It doesn't mean that clock and cycle directly regulate them by binding to them, but they do have binding sites. And so a recent paper that just came out last month, which is a fancy new technology that actually allows you to look at the whole genome in one experiment, looking where DNA binding proteins bind. And in fact, the vast majority of these genes which have clock and cycle binding sites and oscillate are directly bound by clock and cycle, which means that the central oscillator in the organism is directly regulating more than half of our genes. Now, the one thing it doesn't tell us is, well, just because it oscillates, does that mean that the oscillation is significant for our biology? Well, technically no, because it could be that it oscillates, but it never gets below the level that is important for some biological process. So there's more to be understood regarding whether this oscillation has a significant impact on whatever that gene is needed for. But it's stunning as a result to see that there is potentially regulation of a vast fraction of our genome by the central oscillator. That was very simple mechanism with lots of little bells and whistles that we didn't have time to talk about.
The Origin Science Scholars Lectures are presented by Case Western Reserve University's Institute for the Science of Origins, with the assistance of the Siegel Lifelong Learning Program, the College of Arts and Sciences, and MediaVision. For more information on the Origin Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.